Okay, great. Well, thank you very much to Stuart and UK NEQAS for the invitation to speak today, and thank you also to Professor Morley for that interesting talk on some of the history of digital PCR and MRD testing. So I'd like to talk to you today about nucleic acid metrology and its role in potentially supporting the standardisation of MRD testing in leukemia. So as well as looking at sort of future directions for AML, also touching on some of the previous work. So what is metrology? So my husband always says that I have a job title that not even other scientists understand what is meant by nucleic acid metrology. So I think most people would understand the definition of measurement being the process of experimentally obtaining one or more numerical values that can be reasonably attributed to a quantity or property. Well, metrology is the science of measurement and its application. So why does measurement matter? So I'm going to give you an example of something quite far from the MRD application. So going back over 100 years, there was a very large fire in Baltimore, and it destroyed a major part of central Baltimore. And a major reason for the fire's duration was the poor national standards in the firefighting equipment. So although many fire engines came from other cities to help with the fire, many failed to help because they couldn't couple their hoses to the fire hydrants in Baltimore. And the legacy caused a rethink in terms of standardization. And obviously this relates to more standardization of the meter, which is one of the key SI quantities, which I'll come back to later in my talk. So how is this analogous to MRD testing? Well, looking at AML, there's a number of different genetic markers which are used in the monitoring of MRD. And from a sort of diagnostic point of view, we obviously want the accuracy of monitoring MRD with all of these common markers, such as these ones that I've highlighted here, to be as equivalent and as accurate, no matter what the sort of subtype or target that's being monitored. As well, we have a variety of techniques being used for measuring this this quantity, which is central to MRD monitoring, which is that of the target mutation, which is normally normalized to a reference gene or potentially some sort of other reference. And we've got some techniques like real-time qPCR, which has been used certainly for the last 10 to 20 years, where qPCR is initially defining a quantification cycle or CT cycle for both of those targets, and then that's then being used to calculate the copy number concentration from a standard curve. But we also have more recent techniques such as digital PCR and also targeted next generation sequencing, which are also being applied. So we have different techniques here, each of which will have their own sources of error. And it's important that those techniques are also given an equivalent answer for people who are being tested with those different approaches. So I'm going to structure my talk today around some of the three key aspects in the world of metrology and what it really seeks to support. So firstly, I'll be talking about the accuracy of testing, and then I'll be looking at some of the concepts to do with traceability and how that can be support MRD testing. And then finally, I'll talk a bit more about the sort of measurement infrastructure that we have sort of nationally and internationally to support accurate measurements around the world. So starting first of all on accuracy. So accuracy is reflects both the trueness of the measurement, but also the precision of the medicine, the measurement, sorry. And one concept, which again is quite esoteric, I think, to metrology, is the concept of the measurand. So this is the quantity which is intended to be measured. So this really goes back to the actual analyte that is the focus of the measurement. So for example, it could be the ABLE reference gene copy number concentration, which is intended to be measured in the RNA that's been extracted from a sample. So it isn't the fluorescent output from the qPCR machine in terms of the sort of light units, it's the original analyte concentration, and that's important to remember when thinking about how we approach traceability as well in this area. So you can see here that we've got a measurement which is accurate, so it's on the target, and it's also got a defined level of precision. So the measurement uncertainty is an aspect which also is key to measurement science, and something which is now also required as part of ISO 15189 accreditation for clinical laboratories, and ISO 17025 accreditation for testing labs, to be considered in terms of characterizing that dispersion of the values which could be attributed to the measurement that's been performed. And it's normally associated with a particular level of confidence, so where you could expect at a 95% confidence level that all of the possible values would be in terms of around the true value. So here we've got an example of a more precise result, and that would also be associated with a smaller measurement uncertainty. So you're more confident about where the range of values might be. On the other hand, here we have a precise result, but it's a biased result. So it's not overlapping with the actual true value. And also we need to consider, well, does the measurement uncertainty reflect this bias? And there's different approaches to that. But one thing importantly to consider is how do we establish that there is a bias to begin with? How do we validate measurement procedures to make sure they are true as well as precise? So there's different ways of assessing trueness. This could be through reference material, a sample with a known composition. So for a change of, for example, tenfold, then that can be mocked up with samples which are of a known dilution factor. But also reference methods or orthogonal methods can help to validate the intended sort of clinical measurement approach. So some of the key factors, measurands that are as part of MRD testing are the measurement of the target mutation, which is often given in CT or copy number, and that's expressed as a ratio to the reference gene, and then expressed as either a fraction or a percentage. And then in terms of the follow-up, you've also got the comparison between the levels of the target marker at the first time point and then at the second time point. So again, you've got another ratio measurement where the log 10 scale is obviously important for the clinical decision making and diagnosis of whether the disease is progressing or not. And then from a QC point of view, the quantity of the reference gene is also very important in deciding whether the measurement that's being performed is actually conclusive or not. So the CT or the copy number concentration of the reference gene really then reflects whether there's enough cells within the sample that's been taken to be able to give that level of sensitivity that Professor Morley referred to earlier. So yeah, these could be CQ or CT, and also often that's quite then translated to copies as well. Some people may be familiar with this type of fishbone diagram, which is an approach really to mapping sources of measurement uncertainty within a workflow, starting either from the sort of clinical matrix and then looking at some of the pre-analytical factors such as the storage of the sample and the RNA extraction methodology. So again, obviously sort of maximizing the RNA extraction efficiency is going to also give you better sensitivity in terms of detecting MRD, and part of having that reference gene as the control is really reflecting part of that efficiency. Other aspects which I think are particularly important to RNA measurements for MRD monitoring is the reverse transcription efficiency for both the target mutation, which is obviously a lot more variable in AML compared to CML, where there's a predominant gene fusion occurring, and comparing that with the RT efficiency of the reference gene, which is, I think in most cases, independent of the target that's being measured as part of the quantification of the mutant target. So there's many factors to consider and either control or allow for in terms of measurement uncertainty. Moving on to the second aspect I'm going to discuss today is the traceability, or the ability of a measurement to be compared with a standard, or that way of actually comparing different measurement systems such as qPCR, digital PCR, or next generation sequencing based tests for MRD. And this is something which is now part of the newly implemented IVD regulation in Europe, which requires that CE-IVD products have metrological traceability. So what does metrological traceability mean? So this is the property of the end measurement result, whereby the result can be related to a reference through an unbroken chain of calibrations. Each of those is contributing to the overall measurement uncertainty. So it's not just the actual precision or trueness of the result which is contributing to the measurement uncertainty, but also any calibrators which are being applied to standardize the units in which those that measurement is being expressed. So within the IVD regulation, this stipulates that where the performance of devices depend on the use of calibrators or control materials, then the metrological traceability of these values is assigned through a suitable reference measurement procedures or reference materials of higher metrological order. So this really underpins the whole stability of the system, so that any batch-to-batch variation in the manufacturer's diagnostic, or comparing different IVDs for the same indication, the same measurement, are actually comparable. And the ISO standard which deals with this subject of metrological traceability for IVDs is ISO 17511, which was revised in 2020. And this includes a number of example model calibration hierarchies. So these are just a couple of examples here. So the first example deals with systems where there's full traceability to the some of the core SI units, and the SI units which are used most commonly in clinical chemistry are mole and grams. So some of the sort of less complex molecules such as cholesterol and glucose are standardized through a full calibration chain, which is traceable back to SI units. And another example, which my colleagues from NIBSC will be talking about later, is traceability to an international conventional calibrator, such as WHO or international standards, where the traceability is back to that reference material, which allows different IVDs to be calibrated via a calibration factor or conversion factor to the international standard. So returning to the idea of SI traceability, so this really is often described in terms of the SI units, and it's the unit which really realizes what the quantity is that's being described. So if the unit, for example, the mole, is being defined as 6 × 10⁻³ molecules is defined, then all other measurements that are described in moles or molar concentration are then relative to that number of moles as defined through the SI system. And this has been recently revised to include a lot of physical constants actually underpinning the key SI units to really make this a very stable system. And as I mentioned, the amount of substance of the mole or mass in terms of the kilo are the key units really in the area of certainly clinical chemistry. So an example of one of these calibration hierarchies, which is traceable to the SI, would include primary reference measurement procedure, which is often itself then calibrated in terms of using a particular material that has a defined purity and can be weighed out. And that's not often the case for nucleic acid measurements. So we don't often have so much of a material that we can actually weigh it out on a balance and achieve traceability that way. So primary reference measurement procedures are going to be important, I think, in translating some of these approaches into the molecular diagnostic space. So these primary calibrators and primary reference measurement procedures are really an area which is supported by reference laboratories and national measurement institutes like the National Measurement Lab where I work. And these are applied then to the IVD manufacturers' calibration systems and underpin the traceability of the final patient result. So one project which the NML was leading on a few years ago as part of our European metrology funding was BioSI Trace, to try and develop and verify reference methods based on counting or enumeration for biomolecular entities such as lipoprotein cells and DNA. And for DNA, digital PCR was the key reference measurement approach that we were focusing on, as well as looking at NGS as an approach to look at the purity of the materials which are used as standards within that system. So we're looking at the validation and of digital PCR as a technique for realizing this what's regarded as a dimensionless SI quantity of count. So rather than moles, it's about actually counting the molecules, because you don't often know the molecular weight of a piece of DNA or RNA. It can be fragmented, so it's not possible to say in moles how much is there. This builds on some work with other national measurement institutes around the world, where the basis for digital PCR counting was compared with SI traceable approaches, which such as isotope dilution mass spectrometry, which actually hydrolyze the DNA and are then traceable through specific nucleotide standards. And it was found that there was a good agreement between these chemical techniques and the counting-based techniques such as digital PCR, and really laid the foundation then for digital PCR being a candidate primary method. And this is something that we worked on in BioCitTrace to apply to precision medicine, focusing on KRAS SNVs, in particular G12D, and we performed some validation experiments, compared plasmid DNA material with another mass spec type approach, ICP-MS, and that work was published in 2018 to really is a blueprint for trying to validate these primary digital PCR methods, where they have to perform really well, because there's not an approach where you can weigh something out and then have the traceability back to the kilo. And this reference measurement procedure for KRAS P12D was also accepted as the first sort of human genetic reference measurement procedure within the JCTM database. So this is the Joint Committee for Traceability in Laboratory Medicine. And this is a cooperation between the world of metrology, laboratory medicine, and also accreditation, sort of worldwide accreditation bodies. So so ILAC is the International Laboratory Accreditation Committee, and IFCC has that international role in clinical chemistry and laboratory medicine. And the JCTM database has recently just been launched, a new version of that. And that's really an area which underpins a lot of the traceability in clinical chemistry. And as part of my role as a nucleic acid review team leader within JCTLM, I'm keen to have more reference materials and reference methods for molecular analytes included in that database to support standardization in the same sort of way. Just as an aside as well, in terms of the JCTLM, one of the reference materials which is included in the JCTLM database is the ERM AD623 plasmid calibrator, which is something that I think maybe Helen or Nick will touch on later. So this has a defined DNA copy number concentration of BCR-ABL1 and the sort of reference genes used in CML measurement, and that was also value assigned in copy number concentration with by digital PCR, and the NML participated in that a few years ago. So in terms of the infrastructure which supports the activities of metrology labs around the world, so nationally we have the UK National Measurement System, which is a partner within the NHS CSO KTP programme, and examples of the National Measurement System labs includes ourselves, NIBSC and NPL. And then at a European level, we have the Regional Metrology Organisation, which is the Euromet in the case of Europe, and this facilitates funding and joint sort of worked with other European national measurement labs such as BioCitrace, which I mentioned earlier. Now going to move on and talk about some of the international metrology infrastructure as part of our CCQM activities. So as I mentioned before, the SI units are key to international metrology, and that is also the case in terms of the organisation, which is helped by facilitated by the International Office of Weights and Measures, or BIPM, and that organises a number of consultative committees around the SI units. And within the consultative committee for the amount of substance CCQM, there are a number of working groups, including the Nucleic Acids Analysis Working Group and Cell Analysis Working Group. And within the Nucleic Acids Analysis Working Group, we have over 25 members from countries around the world, and we support a range of stakeholder sectors, including feed, food, infectious disease diagnostics, but also precision medicine, biotechnology and gene therapy. And essentially we test the comparability of candidate often primary reference measurement procedures through interlaboratory comparison studies, which benchmark performance and also then lead to calibration and measurement claims which the institutes can claim, and then the evidence for that performance is also kept in a database which is organised by BIPM. So again, it really supports international comparability. And many of the members also produce reference materials which they can then use the reference measurement procedures to apply to these materials. And an example of something we've been doing over the last few years with the pandemic was a pilot study where we were focusing on RNA quantification of genomic RNA targets within SARS-CoV-2. So we had 21 labs participate, and we, similar to the study I mentioned earlier with the DNA measurements, we compared RNA measurements by RT digital PCR with orthogonal methods such as isotope dilution mass spectrometry. And you can see with this graph on the right that for samples that were linked by serial dilution, the RT digital PCR measurements were in good agreement with the orthogonal methods, including IDMS. So this suggests even with the issue of RT efficiency not being 100%, that RT digital PCR is moving to a point where it can be considered a primary method, but we still need to make sure that the RT efficiency doesn't lead to inaccuracy in those primary methods. And another area that we've worked on in the past in the CCQM nucleic acid working group is looking at gene expression measurements which are normalized to reference genes, and we looked at some of the ERCC standards, but also some endogenous genes. And even a few years ago, sort of more in the early days of digital PCR, then we got very good agreement with the RT digital PCR results compared to the assigned values, which also meant this is comparing two different samples, so we were able to dilute the samples by a different amount. So even a few years ago, RT digital PCR was an accurate technique for looking at gene expression changes. So in summary, there's a number of ways which metrology labs can support the standardized measurement of AML, MRD. So one aspect is developing reference measurement procedures to the key molecular targets and mutations which are being monitored in MRD testing. So this can enable traceable values to the calibrating of QC materials which are used in laboratory developed tests or IVDs, enables traceability, and also can provide reference values for EQA schemes to again allow stability within those systems. The development of reference materials is also important, and as sort of mentioned, that with the JRC standards, that can be DNA measurements, DNA standards, which helps support some of the analytical process, but also looking at standards which potentially support the whole workflow, going right back to the actual cells which are being analysed. And this could be something that may be an interesting future direction for joint activity between the CCQM nucleic acid and cell metrology working groups. And joint initiatives such as JCTM can help to really have a references, that all labs around the world can refer to in trying to make measurements comparable. So I'd just like to finish by thanking the members of the CCQM who participated in and helped to coordinate the P199B study, and also for my colleagues at NML and for our funding from BAYES and European metrology funding.